Hello, Great Walls. I hope you're all well. We're going to be talking a bit about genetics and inheritance today. Okay, so genetics and inheritance, what we understand by that is our characteristics, okay, are carried by your genes. Okay, you remember the chromosome from earlier on in the year, right? Chromosome, this is standard chromosome, right? Um, you understand that these are wound strands of DNA that are compressed, right? If you remember, the protein that compresses the DNA into chromosomes are called histones, okay? But that's first term work, right? So this is your centromere, and these are your chromatids, okay? So on your chromatids, they are lokeys, okay? A lokey is the position that your gene occupies, that a single gene occupies, okay? And that occupation of the gene is an allele. So allele, okay? So this allele codes for a specific characteristic. But we'll get into that a bit in a let's just lay the foundation there for a bit. So things like when you have blue eyes, or the length of your hair, or how girthy you are, those are all determined by genetics. Some diseases may be passed down through your genes. So your inheritance is genetics. Genetics is a kind of memory transfer system from one generation to the next. Okay, you could be carrying the nose that had sat on your great 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 grandfather some few hundred years ago, right? And that is possible through genetics and inheritance, right? So genes are inherited through the gonosomes, okay, by gametes. Your gametes are your sex cells. So there's recombination that occurs during meiosis, which allows for there to be a variation in the gene sequence rather. So your specific gene sequence codes for your physical characteristics, and we'll talk about that in a moment. So yes, it is entirely possible for both your parents to be brown-eyed and for you to be blue-eyed. And that is possible through the way that genetics works, and we're going to dig into that now. Okay, so genetics. Just a second. We're going to talk about a genotype, just to lay some terminology foundations here. A genotype and a phenotype. Okay, phenotype. The genotype is chemical in nature, and refers to the genes, the actual genetic sequence of the individual, right? Of the organism. Chemical in nature because you might be thinking, why am I talking about chemicals all of a sudden? Your genes are simply molecules. Okay, DNA, the whole DNA structure is simply specific molecules, right? Phosphate ions, ribose, deoxyribose sugar molecules, and your amino acids, right? Guanine, thymine, cytosine, adenine, right? Uracil in certain cases when we have transcription taking place. So your genetic sequence is entirely chemical. So your genotype is simply chemical. It is what molecules occupy your gene sequence, right? That's the genotype. Phenotype is how the genotype is expressed, okay, in real life. So this refers to the pH here for phenotype is the same as physical characteristics exhibited by the organism, right? The outward characteristics. Now it would be incorrect to say physical appearance. This would be entirely wrong, because it's not just about how an organism looks. Yes, a phenotype is the hair type that you have. It is um, the angular, if you have a sharp jawline, that sort of thing would be correct. Yes, that is your phenotype. But it can also be things like resistance to temperature. Just a list of few examples here for your understanding. It can be resistance to temperature, right? Resistance to a bacterial infection, to microorganisms. It could be your immunity levels, right? For example, certain types of crops, right, display a greater resistance to extreme weather conditions. Yes, that is genetic, but now in genetics we have a genotype and a phenotype. Genetically, yes, they have a certain coding, a certain sequence of gene molecules which allows that resistance to extreme temperatures to be expressed in their phenotype. So yes, genotype, right, codes for the phenotype. So when you make changes to your genotype, you affect the phenotype. It doesn't work the other way around. You cannot change a phenotype and expect a genotype to differ. For example, if you stub your toe, right, say you stubbed it very hard for some reason, you walked into a door, you're very upset at something, and you broke your toe, right, to the point where it then became infected and it had to be amputated. Now you changed your phenotype, right, sort of in quotes, okay. You stubbed your toe, now you no longer have a toe. That's how dramatic it was. Now when you foster children, when you procreate and make children, right, your genetic structure will still continue to code for that lost toe, which you don't exhibit any longer because it was removed. So you affected the phenotype physically, right, from the outside. But the chemical nature of your genes, every somatic cell in your body, still coded for that toe. So all your offspring will be born, yes, with a toe. And that's why phenotype does not affect genotype, but genotype rather influences the phenotype. What your genetic structure is, the specifics of it, in which sequence the genes are placed on your DNA molecule, affects the phenotype that you display, your outward appearance to the world, or your physical characteristics, as you said, your physical characteristics exhibited by the organism. So phenotype affects genotype. Sometimes we can have latent genes in which you require certain environmental conditions, right, to bring about, like you need a trigger for that physical characteristic to show. Okay, for example, heightened stress levels to secrete a certain chemical in the brain, which will allow you to act a certain way or do a certain thing in certain organisms, such as that under this applies more to organisms like that undergo metamorphosis, okay, physical changes to their being as they grow older, right? Like they may lie in a dormant stage, mostly organisms that have life cycles, okay, like your bees, most insects, caterpillars, stuff like that. So they require, for example, very warm conditions in order for them to secrete the chemicals necessary to undergo the biological changes to turn into a butterfly, to start spinning the cocoon, right? So we can also say that genotype plus environment, let me write that a bit neater, environment produces a specific phenotype. Now we can go and stay a step further that the genotype, so your genetic sequence, can still remain dormant even if the environmental conditions are there, but it requires a trigger. So instead of environment, we can say trigger, okay? Because it doesn't necessarily mean, well, it should be the same thing, but trigger would be a better word to describe it. So genotype plus a trigger for those genes gives you your phenotype, but that's a bit into detail, right? Usually, like for a human being, you will carry out your life cycle just based on the unwinding or the expression of your genes, gene expression, just normal. So like you won't look different if it's hotter or colder, you won't retard your growth. So genotype, your phenotype, your outward appearance is dependent entirely on the genes that you have, right? Just I want to establish that connection that we have there, right? And I'm going to show you how, by playing around with the genes, the different gene combinations, we can actually predict what can be inherited. Now remember, these are probabilistic, so we have certain percentages that can happen. So let's talk about why this is in a minute. So we said our allele is basically a spot on the chromosome, right? Okay, it's a very wonky looking chromosome, but a chromosome nonetheless, right? And this is a specific allele which codes for something. Let's say it codes for height. Let's not name it A. I don't know why we did that. Okay. Alright. So we're naming it. Let's say this codes for how tall you are. So height. Okay. We also have a concept of dominance and recessiveness. Okay, so dominant genes dominant genes overpower the other genes. Now, why this happens is a bit obscure, but we think that physically, right, dominant genes are able to attach to chromosomes more freely. They're able to move about during recombination a bit more, and recessive genes are kind of like just going with the flow, just kind of not really there. They can be easily overpowered by the dominant gene. So let's say height, right? So we have a big H, this will stand for dominant, if it's dominant, a dominant gene, right? And a small lowercase, or I should say uppercase. Uppercase refers to dominant, a dominant gene. These are symbols that we use, right, to express them. And I'll show them to you in the examples, and it will make more sense. Dominant gene. And lowercase is recessive. Okay? Uppercase dominant, lowercase recessive. So the allele for height that's on this chromosome, okay, for height, so this would be if height is dominant, right, and if height is recessive. So the type of height, so tallness is dominant over shortness, right? That's scientific. So tallness is dominant over shortness. So let's use the symbol T, the dominant symbol. So this means tall, and because it's an uppercase, it's going to be dominant, right? And this means short. We don't call it T and S, no, we call it T, big T, small t, okay, because it's a recessive version of tallness, right? Recessive tallness is just shortness, right? So this is short, and this is a recessive gene. So your genes, you carry two alleles, one of paternal origin and one of maternal origin, okay? So you have two chromosomes your chromosomes are in pairs. I hope you know that, and one from your mother, one from your father. So you have two alleles coding for the same genetic characteristic, for the same phenotype, right? So like your eye color will determine on your mother's phenotype as well as your father's phenotype, and whichever is dominant, that's what you will show. Okay, so if your mother had brown eyes but your father had green eyes, so you have big B and small b, okay, let's call them brown and green, because and green is recessive, brown is dominant, okay, you will be brown, you will have brown eyes, okay. If both your parents carried brown eyes or green eyes, you would be green eyed, but we'll show you how that works. Yes, you still can be, depending on words like homozygous and heterozygous. Okay, we'll look at that with an example. I think that would be much better. Okay, so let's just cover that. Homozygous and heterozygous. What do we mean by hetero and homozygous? Homozygous refers to being the same, right? The same. Just that's all. Just don't think too much. Just the same. And heterozygous is different. Okay, homo means the same, hetero is obviously different. So the same meaning. So say I tell you that your mother, right, your mother, this woman here, is heterozygous for having brown eyes. So we know that the symbol for brown eyes is a big B, and because she's heterozygous for it, we have two big B's there. Okay, and if we said that she was homozygous for brown eyes, one allele is dominant but the other is recessive. Okay, so they are giving you all the information, you just need to understand what these terms in fact mean. Okay, now this is a very interesting process called genetic crossing. Okay, and it was done by Gregor Mendel. I hope you know this man. He's a very prominent figure, the father of genetics. Gregor Mendel, right? In about the late 1800s, right? His first official publication, I think, was in 1900. So Gregor Mendel, he was an Austrian monk. He lived in a monastery and he liked playing around with pea plants, right? So he took thousands of pea plants and just crossed them. Like, it was a lot of work because he had to make sure that they weren't crossbred, or he had to make sure that they pollinated in the correct sequence with the correct characteristic, and then he tabulated it and got his results. And yeah, so what he said basically, he explained to explain okay, actually let me walk you through Gregor's process, and then you can have a better understanding of what exactly is going down. Okay, so he crossed parents which differed in respect of chosen characteristics. Okay, so tall or short, right? Red or white flowers, seed colors, yellow or green seeds, okay, smooth or wrinkled seeds, that kind of thing. Okay, so he contrasted characteristics um, for the same feature. So if he took, for example, flower color, he took red and white, and he would assess which one is dominant and recessive by conducting his experiments, right? So he planted, so he did these crosses in the F1 generation, stands for first filial generation. First filial, F stands for filial. Okay, so that's an F, filial generation, right? And then afterwards you get the P generation, okay, parent one, parent two, and stuff like that, okay. And sorry, he crossed the initial parent one, parent two, etc. First generation of parents, and then he obtained a first filial generation, okay, meaning the products of the parents, right? And he recorded the appearance and worked backwards as to what the genotype would have been, that sort of thing, because he couldn't exactly go and find out the genes, because genetic testing was not that advanced 200 years ago, right? It simply wasn't. So he couldn't immediately tell the genotypes of the plants, so he had to tell the phenotypes. Well, that's observable, right? Hence the word observable is in the definition here. So observable physical characteristics exhibited by the organism, that's important, right? Then what he did, he allowed these offsprings to self-pollinate and proceed to form cross-pollination by bagging. So he bagged, that just means that he bagged the flowers so that they can only be pollinated by themselves. Okay, he planted the seeds that he got from this. It was a very long process, and he obtained second filial generations, third filial generations, did his tabulations, all the statistics. Yes, it's a very, very um, hectic process that he went, but he had time, right? He was a monk. There wasn't really much going on in his life. Do you know he actually wanted to be a teacher, but he failed. He failed the entrance exams. Yeah. So his experiments involved. This is an important point. Okay, a single pair of contrasting characteristics. These were called mono hybrid crosses. Mono hybrid crossing. Okay, when there's a single pair of contrasting characteristics, that means the same thing. If you're taking a seed, you're gonna contrast the wrinkled seeds and smooth seeds. Okay, if you're taking hair color, oh sorry, hair type, you'll take short hair or long hair. Okay, smooth hair or frizzy hair, right? If you're talking about your ears, lobed or unlobed. All right, there's a whole bunch of things, whether you can roll your tongue or whether you can't roll your tongue. Single pairs of contrasting characteristics. Mono hybrid, so single being mono, hybrid crossing, so single pair of contrasting characteristics, right? So let me show you his results in a second. Yes. It's a bit complex, so let's not go into Mendel's result. Let's just go into an example. So he explained that gametes of red flowered plants, right, would contain a factor for redness, which is responsible for the characteristic that the plant became red with the flower, right? And then the gametes of the white flowered plants contained a factor for whiteness, right? These factors that we're talking about became later on known as alleles, okay? That thing, this indiscriminate thing, indiscriminate being it can't be broken down further, which codes for a characteristic, okay? That factor of redness. So you have a factor for tallness if you're tall. You have a factor for frizzy hair if your hair is frizzy. That factor is an allele, right? That's just what I want to explain, which allows your hair to become frizzy because it is encoded in your genes, right? But then now, when you cross a red flowering plant and a white flowering plant, okay, a red flowering plant and a white. So let's white flowers. That's a flower. Just don't judge me. And a red flower. Okay, you cross them, and you happen to get red flowers. So the factor for whiteness in this one's allele and the factor for redness in this one's allele must both be contained in all of the offspring. You must have each a factor of whiteness here from this parent, because remember, one set of chromosomes, so it's getting this allele and it's getting this allele, right? So it's also getting the red allele. So that's what the genes look like. But because the red is exhibited, is observable, right? Red is the phenotype. The phenotype is red. This is the genotype. They are red and white. So this is only white. Purebred, that's what that means when it's homozygous. Homozygous is also purebred. Okay, so homozygous, let's write that in here. It's a little mixed up as I'm showing it to you now, but you'll see the bigger picture once we do an example. Okay, so this is homozygous, right? And these become heterozygous, because they have contrasting, or they have different alleles. Look at that, different alleles, but they contain a factor for whiteness. It contains a factor for redness each, but the redness is showing. What that means is that the redness has to be dominant. So this has to be a dominant trait. Redness is a dominant trait. That's the kind of things that Mendel was able to observe. Okay. So I hope that makes sense to you, right? So let's do an example. Okay, let's do an example. Let's say, I'm going to write this out. Crossed a homozygous red flowering plants is crossed with a homozygous white flowering plants. Determine the possible offspring if red flowering, if the red flowers are dominant, if red is dominant. Okay, so this is the information they'll give you. They'll give you information about the parents, and you'll be asked to work it out. So yes, it is a sort of working, not exactly a calculation, but it is a working nonetheless. Okay, so we start with our first parental generation. Okay, so P1, first parental generation, P1. This is exactly how you lay out your working. Let's put in some lines, because this is, yes, exactly how you will set it up in the exams. Okay, right, so we get P1. Let's zoom in there. P1, and we show phenotype first. It's phenotype first. Phenotype first. Yes, that alliterates for a reason. PHF, phenotype first. You write phenotype, yes. You write genotype. This is my method, genotype. Well, just writing most of it first. It's the accepted method in the memos. I didn't make up the method, it's just that this is how I do the method. So we stay there. It's a red crossed with white. So you write down what the characteristic is. Okay? Now the red will come from, let's say, the male, and the white comes from the female. Okay? Right. I think those symbols are correct. Am I mixing them up? Yeah, the red comes from the female, the white comes from the male. All right. And we have the genotype. So what would the genotype of a homozygous red flowering plant would be? Capital R, capital R. Okay? The genotype for a white flowering plant would be small r, because it's the same characteristic, monohybrid. So the same characteristic is the color of plants. If red is dominant, we use the symbol R. Big R for the dominant one and small r for the recessive one. So don't put small w. Okay, that's not going to make sense. And then we have our gametes. So after meiosis will happen, so actually let's write there meiosis happens. Sometimes it's a mark. Meiosis takes place here. Okay, and that means we are left with 4 gametes. Okay, so we have a recessive, so in the one cell produced from the mother, right, we can get a a red flowering plant gene and red flowering plant gene. Those are the only possibilities we can get, nothing else. And we can only get white from here and white from here. We can't get, there are no other possibilities, right, in our daughter cells. And yes, why don't we show like 4 different things, because that's accurate? No, no, no. We only show 2 because one sperm cell, one egg cell, right? So one sperm cell will have 2 characteristics, one egg cell will have, and so forth, okay? I'm not quite, will have 1 each, but that's not the point, okay? These are your gametes, so you put their gametes. And then you put fertilization. Because fertilization now happens, doesn't it? And now at this point we can use a Punnett square or crossing. Now I prefer using the Punnett square because it is a set method. So let's put our female gametes here. Okay, and our small r there, right? It's Punnett square. Now how we fill this in is that we just multiply, okay? R here, R there, R there, R there. Now from the top, small r, small r, small r, small r. So that's what our offspring will look like. So then we put F1 generation, the first filial generation, right? Now we put genotype because we need to list our genotype, and we can just write here all RR, okay? And then our phenotype, we extrapolate the phenotype from the genes. All red flowering plants. All will be red flowering. Okay? So every single offspring that this parents will produce, the red flowering, the white flowering, will be red flowering because of the red being dominant, okay, and it being homozygous. Now let's say that the plants of the F1 generation were crossed. Determine the possible phenotypes of the offspring, right? This is a nice one. Let's have a go. So remember the system, we say P1, okay, phenotypes, or just phenotype. We have red, and we have the red heterozygous. Okay, if you want, you can write in brackets heterozygous if it makes you feel better. I think it is a good thing to do. And here we'll write crossed with red heterozygous. So we know that there's a dominant gene and a recessive gene. So dominant and recessive alleles exist within this heterozygous. Lovely. Now our genotype. The genes for this, because it's heterozygous, will be R and R, R and R, right? Meiosis happens. Okay. Meiosis, okay. Then our gametes will look like this. We'll get 1 gamete from here. So remember what happens in meiosis, okay? Your sperm cell will either have because a sperm cell or egg cell, right, has only 23 of, sorry, not plants, will have half the number of chromosomes. They are haploid, right? Haploid. Because they're haploid, they only contain 1 set of chromosomes. So they'll either contain the allele with red, the dominant one, or they'll contain the allele with white, the recessive one. Okay, so we have to include both options. Remember, as I said, that this is probabilistic. That's why. Because we don't know. It can be either or. So either we have red, or we can have a sperm cell that has the small r. Same on this side, right? Not always the same. We could have crossed it with a homozygous RR and saw what happens. So the question could have said one of this F1 generation was crossed back with one of the P1 generations. Show that. That's what we can do next, right? So RR times big R, small r. Okay, and then we have fertilization. Okay, please write it in full in the exams. Fertilization happens. Okay, so let's do our Punnett square. I think it's a more reliable method than the other ones. So R, 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 R. So figure out a system for you to do it on your own, like an algorithm. It's a very fun, very practical thing to do for a lot of marks also, right? And yeah, so let's go top. So that's R, R, R. Now let's go left to right. Another R. Oof. Remember, your small letter goes to the left. So that's, sorry. It's going to be another small R there, right? So these are all the possible offsprings. Interesting, isn't it? Interesting. So let's write out. First, we state that this is the F1 generation, or else it's going to be F2 generation, isn't it? P2, F2, right? So this is our second parental generations, because it's following on from here. So this F1 first filial generation becomes the first parental generation in this cross, and what follows is the second filial generation, right? Just hope that makes sense to you. Let's fill this in. So we're going to say genotype first here because we're writing what's in the Punnett square. Okay, Punnett square is used in mathematics also when you're learning, I think, in grade 2 or grade 3, and then phenotypes. Do this neatly, do this slowly, make sure you know what you're doing, alright? And yeah, you can also put arrows. I like putting arrows showing where my gametes came from. Oh, sorry, I didn't put R here. I like to show where my gametes came from, okay? So the genotype here would be RR, list them. Then we get RR, get RR, and lastly we get small r, small r. So the phenotypes are going to be dominant red. So you're going to say homozygous red, right, is to 2 heterozygous red is to 1 homozygous white, right? If you want to know what proportion of the offspring will be red, you will say 3 is to 1, okay? Because all of these will be red. So we just specified which is homo, which is hetero, and I think you should as well, unless they ask for other things. If they ask you what proportion will carry the white allele, 3 is to 1, right? If they ask you what proportion would be white, 1 is to 3, okay, or 25% rather, right? That's easy to see. 50% is heterozygous red, and 75% is simply red. So depending on what they're asking for, that's how you will state it, okay? Now let's say let's say a homozygous white is crossed with a first filial generation. Sorry, one of the first filial generation plants. So we're gonna say let's start a new one. So we say P1, okay, phenotype, genotype. Okay, so the phenotype is gonna be homozygous white and heterozygous red. Cross. Got it. And the relevant genotype is going to be, you can be as extra as you want with this. Make it neat, make it fancy, figure out your own type of style, but make sure that the information, the key information that is there, follow that algorithm. Phenotype, genotype, meiosis, gametes, fertilization, genotype and phenotype again, right? So it's like a sandwich kind of thing that you need to show, starting with phenotype, ending with phenotype. The genotype is then going to be RR times RR. Got it. Gametes. First, we show that meiosis occurs. Okay, it's a very important, okay. Meiosis is occurring here. Then we have gametes. What are the gametes? So we get one gamete that could be small r, the other gamete will also only be small r. Then we get one gamete being big R, another gamete also being small. If you like probability in school, you'll enjoy this. Your tree diagrams, okay? Because it's the same principle that's going on here. Okay, gametes. Then we show that fertilization occurs. Fertilization. I like to distinguish my processes from my statements of phenotype, genotype. So, like you can do this thing here where you put a cloud, or you can underline it to put in different color. All of that, just to make it stand out so you know what you're doing. It's very important not to confuse yourself. You can get easily confused, especially when we work with dihybrid crosses and stuff like that, right? So that's that. Now, we do the Punnett square thing, okay? The other method is to do this. So you say one red from here, one from there. It's going to be big R, small r. We can go from here and there, and we can say, yeah, there, and then this one here. And that's going to be r as well. Then we say this one, but with this one is now going to be r, and then this one with this one is going to be r. But that gets a bit confusing, and you need to do it carefully. So let's do our trusty Punnett square, r. I keep saying the same r often. I hope you know I mean different r's, and it is evident, so you should know I mean different r's, right? So then we have big R, small r, we have big R, small r, small r, small r, small r, right? Now in this case, what's happening? Interesting, isn't it? So this is our first folial generation because we restarted the thing. And we're going to call this our genotypes first because we need to list what's there, right? Genotypes. Okay. Oh, we don't cloud that one. Okay, our genotypes are R is to R. I reckon that's easier than writing it out 4 times, isn't it? Because the proportion is the same. So 50% will be heterozygous red, 50% will be homozygous white. So we write out our phenotypes, and we're going to get, red, heterozygous red is to white in their exact proportion, okay? We don't need to say homozygous white because it's implied that white is homozygous by the fact that it is recessive. So white can only be shown in the phenotype if there are two of it, if it is homozygous, okay? So just make a note that recessive alleles must be homozygous to be expressed in the phenotype, okay? Okay, so this is also called your phenotypic ratio, by the way, 1:1, okay? And that's all for now. In the next one, we will look at other types of genetic crossing.